Good morning. I am Josephine Alcantara of Caloocan City Business High School. My art attack is the material for genetic engineering topic. Many years ago, diabetics who needed insulin to survive had to inject cow and pig insulin into their bodies. This was not ideal for many reasons. Today, the insulin that diabetic inject is human insulin, and it is produced by microorganisms such as the E. coli bacterium and certain strains of yeast. But how do these microorganisms produce human insulin? Genetic engineering. Scientists are able to turn certain microorganisms into mini factories that make useful substances that improve our health, the environment, and the economy. But what is genetic engineering? An organism's DNA makes up its genes, which code all the proteins the organism needs to survive. Each gene codes for different protein or part of a protein. Genetic engineering is the manipulation or changing of the DNA of an organism. Genetic engineering involves removing a gene from one organism, which is called the donor, and transferring it to another organism, which is referred to as the recipient. The recipient is called the transgenic organism or genetically modified organism. There are two purposes of genetic engineering. Sometimes we require large volumes of particular protein to be made. Scientists use transgenic microorganisms to produce large volumes of protein, for example, insulin growth hormone or vaccines. An organism B can have a gene from a different organism and introduced to give it to some advantage that has naturally. Organisms like B are called genetically modified or transgenic organism. For example, a gene that manufactures a toxic chemical in a bacterium can be introduced into a maize plant to make a plant toxic to a certain insects that would eat, such as caterpillars. Now let's look at the processes of genetic engineering. How do you transfer a genetic characteristic from one organism to another? Although it's a very complex process, it can be summarized rather simply. Well, use the example of how the insulin gene is inserted into a bacterium in order to manufacture large amounts of human insulin. The gene that codes for human insulin is found at chromosomes 11 at position 15.5. The insulin gene that cut from the chromosomes 11 using a special enzyme called restriction enzymes, bacteria possesses small circular strands of DNA floating in their cytoplasm. These strands of DNA are called plasmid. Plasmid is extracted from the bacterium cell. The plasmid is cut open using the same restriction enzymes that were used to cut the insulin gene from the human chromosome. The restriction enzymes leave sticky ends where one of the two DNA strands is slightly longer than the other, because the same restriction enzyme was used to cut both the human DNA and the bacterial plasmid. The bacterial plasmid, the sticky ends are complementary and will allow for joining using the complementary base pairing. The insulin gene is joined to the plasmid by the sticky ends. The enzyme called ligase is used to complete the joining of the two pieces of DNA. The modified plasmid is then inserted into the bacterial cell. The modified bacterium is genetically modified or transgenic organism. because it contains some human DNA along with its own bacterial DNA. Its bacterial DNA is called recombinant DNA. Bacterial DNA lined with human DNA. The bacterial cell is placed in the fermenter, which allows for rapid asexual reproduction. Ideal conditions with the optimal temperature, pH, and lots of food. Because the bacteria reproduce asexually, the offspring are all clones of the original transgenic bacterium. They all have identical recombinant DNA. The bacteria express their own genes in order to survive. In other words, they make their normal bacterial proteins, but they also express the human insulin gene and make human insulin. The fermenter has become a culture of many factories all producing human insulin. The insulin is then extracted, purified, and packaged. Humans can now inject themselves with human insulin, and that is how the genetic engineering works.